Welcome to the deep dive. Today we're jumping into something really fascinating, alien life. But maybe not where you'd first think. Our mission today, to understand how scientists are, well, literally creating new kinds of life in the lab and, what that really means for all of us.
Yeah, it's interesting. When most people hear xenobiology, they probably think sci-fi, right? Searching for life out there in space, what we'd now usually call astrobiology or exobiology. The name comes from the Greek xenos, meaning stranger or alien. And for a long time, people kind of joked it was a science in search of a subject.
Right, so it's not just a small tweak in meaning. This sounds like a huge shift, a radical rethinking of life itself, really, driven by what we can now do.
Exactly. The definition of xenobiology, the modern one, has completely changed. It's now very much an active engineering field, part of synthetic biology. So instead of just looking for alien life, modern xenobiology is about designing and building biological systems, ones with totally different biochemistries, things you just don't find in nature. We've kind of gone from searching for aliens to being able to, well, create alien life right here by rewriting its basic chemistry.
Okay, that distinction is crucial. Astrobiology looks, xenobiology builds.
That's the core difference.
So if we're building life now, why? I mean, why pour so much effort into something that sounds so incredibly ambitious? What are the main goals?
Good question. There are really three big motivations driving this. First, it's about digging into fundamental knowledge. Pure science, you could say. By actually building working biological systems with different chemistries, maybe using different genetic molecules or building blocks for proteins, researchers can test the limits, the actual constraints that shaped life as we know it here on Earth. It shifts the study of life's origins from just looking back historically to actually experimenting.
So it's less about finding what is out there and more about testing what could be possible for life. Like, are Earth's rules the only rules?
You've got it. That's a big part of it. Second, there's the goal of engineering novel technologies. Think about developing new industrial platforms or medical ones with totally new abilities, like creating new kinds of materials or proteins that do things natural proteins can't because we're using some of the hundreds of other amino acids nature doesn't typically use, or even building super efficient cellular factories for making things sustainably.
Okay, practical applications. Makes sense. What's the third one?
The third one is, well, potentially huge for safety. It's about creating the ultimate biocontainment system. They sometimes call it the genetic firewall.
Genetic firewall. How does that work?
Okay, so imagine you build an organism with a fundamentally different genetic code and biochemistry. It's so different that swapping genetic info with natural life forms is just chemically impossible. They can't understand each other. Plus, these organisms would absolutely depend on synthetic building blocks, things you have to supply in the lab because they don't exist outside it.
So if it escaped, it couldn't survive or reproduce because its food isn't available in the wild.
Exactly. It's fundamentally dependent, like a car that only runs on a fuel you can't buy anywhere.
Yeah.
They've actually built organisms like this genomically recoded organisms, or GROs, and they've shown they have incredibly low escape frequencies. We're talking like less than 1 in 10 trillion, so it's a powerful safety concept.
Wow, that's impressive containment. But it does make me wonder, how? How are they actually doing this at the molecular level? What's the tech?
Right, the how. This is where it gets really clever. They're focusing on two main areas. First is xenonucleic acids, or XNAs. Basically, they replace the natural sugar backbone of DNA or RNA, the rails of the latter, with different chemical structures. The rungs, the information-carrying bases like A, T, C, G often stay the same, but the backbone is totally artificial.
So they're changing the paper the genetic code is written on, not necessarily the letters.
That's a good analogy, yeah. And they've shown these XNAs can actually store genetic information, pass it on, they can even evolve in the lab. It proves heredity isn't exclusive to DNA and RNA. Plus, natural enzymes usually can't break them down, which makes them really interesting for things like stable drugs or diagnostics.
Okay, X and A's. What's the second pillar?
The second big push is expanding the genetic alphabet itself. So instead of just the 4 natural letters A, T, C, G, the goal is to add a third synthetic unnatural base pair. Let's call it X and Y. The big breakthrough here came from Floyd Rumsberg's lab. They created the first semi-synthetic organism, an SSO. It's an engineered E. coli bacterium that can hold onto and even copy DNA containing this third unnatural base pair. So it's basically the first stable life form with a 6-letter genetic alphabet.
A 6-letter alphabet. That really does feel like rewriting life.
Yeah.
So stepping back, what does all this mean for us? Where's this heading?
Well, the implications are pretty profound and honestly a bit complex. You have this paradox with containment, for instance. The genetic firewall is strong, yes, but the more alien, the more biologically different and successful a xenobiological organism becomes, the harder it is to predict what would happen if containment did fail, however unlikely. It shifts the risk from genetic pollution, which we sort of understand, to ecological pollution, which is much more unknown territory.
A whole new kind of ecological unknown.
Exactly. And then there's what people call the ELSI lag. ELSI stands for ethical, legal, and social implications. Basically, the technology is moving incredibly fast, but our thinking about the ethics, the laws, the societal impact, it's lagging behind. Xenobiology forces us to ask really fundamental questions. What even is life? What's our role as creators, potentially?
Deep questions indeed. And looking further ahead.
Looking ahead, you're almost certainly going to see more convergence with artificial intelligence. AI can potentially design totally new XNAs, new unnatural base pairs, new proteins much faster than humans could, which is exciting but also adds another layer of complexity. If AI designs something in a black box process that we don't fully understand, how do we effectively assess the risks? How do we ensure safety?
Right. AI is a partner in creation, speeding things up but maybe making them harder to fully grasp.
And definitely complicates the picture.
So it leaves us with a pretty big thought, doesn't it? What does it truly mean when humanity, maybe partnered with AI, starts designing life in ways nature never did? Something to really mull over.